Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Imagine something you might see swimming around in a home aquarium. A tiny striped zebrafish.
Mm-hmm.
Now, picture that same little fish holding a secret, maybe a crucial piece of the puzzle for understanding and, well, possibly even treating an incredibly rare, really devastating genetic disease.
That's a fascinating picture.
Yeah, and that's the intersection we're diving into today. We're going to explore how researchers turned to this, well, unexpected model organism to make a significant leap forward.
A leap in finding potential therapies for an ultra-rare muscle disorder called X-linked myopathy with excessive autophagy. Quite a mouthful.
It really is. And you said ultra rare. That's not an exaggeration here, is it?
Not at all. X-linked myopathy with excessive autophagy, it's a severe inherited condition. Primarily causes progressive muscle weakness, but the sources show it can hit other organs too.
Okay.
And its rarity is it's one of the biggest hurdles. As of early 2024, there were only something like 33 known cases reported worldwide.
33 globally. Wow. That number just really hammers home the desperate need for any kind of progress.
Absolutely.
So for this deep dive, we're pulling insights from a health news article that gave a good summary, but we're really going to dig into the details from the scientific paper itself. It was published in Embio Molecular Medicine.
Right. And our mission here is really to walk you through this remarkable study. We want to unpack, why zebrafish? Why these specific fish?
Yeah, what did they find out about the disease using them?
Exactly. And then the drug candidates, they stumbled upon some surprising ones there, but also, importantly, the limitations. What are the caveats when you try to translate findings from fish to humans?
Right, that's key.
And ultimately, what could this all mean for treatments down the road for patients living with this condition?
Okay, sounds good. Let's start with the disease itself, X-linked myopathy with excessive autophagy. Beyond that really long name, what's actually happening inside the body? What's going wrong?
Okay, so at its core, it's this rare inherited muscle weakness disease, usually starts in childhood, and it's X-linked, which explains why it predominantly affects males.
Got it.
The root cause. It's a mutation in one specific gene. It's called VMA21.
VMA21. And what's that gene supposed to do normally?
Well, it plays a really, really critical role. It helps assemble this vital protein complex inside our cell.
A complex, like a little machine.
Kinda, yeah. It's called the vascular hydrogen ion ATPase. Think of it as a tiny essential pump. Its whole job is to pump hydrogen ions, basically, making things acidic into these compartments in the cell called lysosomes.
Lysosomes, okay, I think I remember those. Aren't they often called the cell's recycling centers?
Yeah.
Where old stuff gets broken down?
Exactly. That's a perfect way to put it. Lysosomes are the cleanup crew. They handle breaking down damaged proteins, old cell parts, general waste, through a process called autophagy.
Autophagy, right.
But here's the catch. For that breakdown and recycling to work properly, the lysosomes absolutely need to be acidic. And that pump, the vascular hydrogen ion ATPase, which needs the VMA21 gene product to get assembled correctly, that's what makes them acidic.
Okay, so let me see if I've got this straight. If the VMA21 gene is mutated, then the pump doesn't get built right or doesn't work correctly, so the lysosomes don't become acidic.
Precisely.
And the whole recycling system, autophagy,
Yeah.
it just jams up. Grinds to a halt.
You nailed it. The process, sometimes called autophagic flux, the flow of material through the system gets disrupted. Instead of waste getting broken down and cleared out, it just starts to accumulate, especially in muscle cells.
And that's where those vacuoles come in, the things they see under the microscope.
Exactly. That buildup manifests as these characteristic fluid-filled enclosed structures, the vacuoles, inside the muscle cells. It's a classic sign of the disease.
The name excessive autophagy feels a little counterintuitive then, if the process isn't actually completing and things are building up.
Yeah.
Is it maybe more about the failure of autophagy leading to an excess of accumulated stuff?
That's a really sharp observation. And yeah, it highlights a bit of nuance. While the name stuck, the actual cell biology findings, including in this study, strongly point to a disruption or a blockage in the flow, the later stages of autophagy.
Yeah.
It's not necessarily that the initial steps are hyperactive, it's that the final breakdown in the non-acidic lysosome just isn't happening efficiently. So, yeah, disrupted process leading to excess accumulation is probably a more accurate picture of what's happening at the cellular level.
Got it. That makes more sense. And the sources mention that while it's sometimes described as, mild to moderate, the reality is quite varied. Some cases are extremely severe.
Absolutely. The clinical spectrum is wide. And this leads back to a major roadblock for researchers. Understanding it better and finding treatments has been really hampered by the lack of good animal models that truly mimic the human disease.
Right. You can't easily study this kind of complex cellular breakdown or test drugs directly in the few human patients there are.
Exactly. Especially with only 33 known cases worldwide, you need something else.
Which, I guess, brings us circling back to the zebrafish, the unexpected hero. If the usual models, like mice maybe, weren't cutting it, why fish? It doesn't immediately leap out as the go-to for muscle disease research.
Right. But that's what makes this work so clever and frankly exciting. While mouse models are often the standard, the zebrafish, Daniarurio, it just offers this unique package of advantages, especially for genetic studies and drug screening like this.
Okay, so what are these advantages? What makes them so special for this?
Well, for one, they develop incredibly quickly. You go from a fertilized egg to a larva with developed organs in just a few days. It's amazing.
Wow, days.
And they produce massive numbers of offspring, huge clutch sizes. A single pair can give you hundreds of embryos at once.
Okay, fast development, tons of offspring. That sounds like a recipe for high-throughput screening, right?
Yeah.
Testing lots of things quickly.
Precisely. And on top of that, they're relatively easy to manipulate genetically. Scientists have really refined tools like CRISPR to make specific changes to their genes very efficiently.
CRISPR. We hear a lot about that.
Yeah, and here's maybe the coolest part for this kind of work. The larvae are transparent, completely see-through.
Wait, really?
Yeah, you can literally put a live fish larva under a microscope and watch its internal organs, watch cells, see processes happening in real time.
That's unbelievable. So you could potentially see the disease effects directly.
You got it. And even though it's a fish, genetically, they're surprisingly similar to humans in many ways, including having many of the key genes involved in that autophagy pathway we talked about. Their muscle physiology also shares important similarities with ours. And sometimes, as these researchers pointed out, zebrafish models can actually reflect the severity of certain human muscle disorders even better than, say, mouse models might.
Okay, wow. So that combination speed, numbers, genetics, transparency, relevant biology, it makes a really strong case.
Yeah.
So how did they actually make zebrafish that had this X-linked myopathy?
They use that gene editing tool we mentioned, CRISPR-Cas9. You can think of it like super precise molecular scissors. They used it to target and cut the zebrafish version of the human VMA21 gene. The fish gene is called VMA21.
So they basically engineered the fish to have the same kind of genetic defect that causes the disease in humans.
Essentially, yes. They created two different types of mutations, both designed to be loss of function, meaning they'd knock out or severely reduce the normal function of the fish's VMA21 gene. This gave them a living model system to study what happens when that gene doesn't work.
Okay, so they created these mutant zebrafish. And the big question, did they actually show signs of the disease? Did they get sick in ways that looked like the human condition?
They absolutely did. It was quite striking, actually. These mutant fish showed symptoms that mirrored many aspects of human X-linked myopathy with excessive autophagy, especially leaning towards the more severe end of what's seen in people.
How do you even tell if a tiny fish larva have muscle weakness?
Yeah, it sounds tricky, but they have established ways. They looked at physical things, like the mutant fish were shorter in body length. They had problems inflating their swim bladders, which affects buoyancy, how they float. And behaviorally, their swimming was impaired. They were less likely to dart away from a stimulus.
Ah, a reflex test, basically.
Kinda, yeah. They spent less time actively swimming overall and covered less distance when they did swim compared to normal, healthy fish. Plus, they used specialized microscopy techniques like birefringence to look directly at the muscle tissue structure, and they saw clear abnormalities.
And what about inside the cells? Did they see the hallmarks, the non-acidic lysosomes, the waste buildup, those vacuoles?
Yes, absolutely. They confirmed that core cellular problem. Just like in humans with the disease, the lysosomes in these mutant fish failed to acidify properly.
Wow.
They also found lower levels of a key protein usually found in lysosomes called lysosome-associated membrane protein 1. And using powerful electron microscopes, they could see those characteristic vacuoles, the fluid-filled sacs, accumulating within the muscle cell.
So you could literally see the recycling system failing at the microscopic level.
Exactly. And they backed this up by looking at other markers related to autophagy, like levels of a protein called LC3, and by using fluorescent tags to track the flow of material through the pathway. All the signs pointed to autophagy being seriously disrupted.
The sources also mentioned it wasn't just muscle, right? There were issues in other organs.
That's right, and this was important because it also mirrored findings in some human patients. The mutant fish developed fatty livers, hepatic lipid accumulation is the term.
Okay.
Their livers were smaller than normal, and they had problems with bile flow, suggesting liver dysfunction. The paper also noted some heart issues.
So this little fish model really did capture multiple key features of the human disease across different body systems. But you mentioned earlier there was a key difference in severity.
Yes, and this is really crucial for interpreting the results. While the human disease is often progressive, sometimes slower, sometimes faster, these mutant zebrafish had a dramatically shortened lifespan.
How short?
None of them survived beyond 12 days after fertilization. 0.
Wow. That's incredibly severe compared to most human patients.
It is. But tragically, it does align with the rarest, most severe cases in humans where infants or young children die very early.
Okay.
The researchers think this major difference in severity might be because the specific CRISPR mutations they created likely caused a near total loss of VMA21 function in the fish. Many human patients might have mutations that still allow for a tiny bit of residual protein function, which could explain the generally slower progression.
Right, that makes sense. But even with that difference, just having a model system that shows the core cellular problems and, crucially, could be used for testing, that feels like a massive step forward, especially since, as you said, there are currently no targeted therapies.
Exactly. This was the breakthrough. Now they had this tool, this living system where the disease process was happening, and they could start systematically searching for potential drugs, seeing if anything could actually counteract the effects.
So where did they start looking? What kind of drugs did they test?
Well, given that the fundamental issue is this blocked autophagy pathway leading to buildup, they logically focused on compounds known to affect or modulate autophagy in some way. They screened a library of 30 different drugs. Importantly, these were drugs that had already been tested in humans for other conditions, so they had some known properties.
Okay, 30 existing drugs tested on these tiny mutant fish. How did they know if a drug was actually helping?
They used a couple of main readouts. They looked for improvements in that muscle structure using the birefringence technique again.
Right.
And probably most critically, they looked for improved survival. Could any of these drugs extend the lifespan of these severely affected mutant fish compared to untreated ones?
Makes sense. Survival's a pretty clear indicator. Did they find anything promising out of those 30?
They did. They got 9 initial hits compounds that showed some positive effect on either muscle structure or short-term survival.
9 out of 30.
Okay. So they took those 9 promising candidates and put them through more rigorous, longer-term testing. They looked for sustained improvements in survival and in the fish's swimming behavior, that motor function aspect.
And did any stand out from that group?
Yes, this is where it got really interesting. 2 drugs emerged as showing the most significant therapeutic benefits in this zebrafish model. Edaravone and another one called LY294002.
Edaravone and LY294000. What kind of improvements did they actually see with those 2? Were they substantial?
They were quite compelling, especially considering how severe the baseline condition was in these fish. Treatment with these drugs significantly boosted survival rates.
How significantly?
Well, get this. For Edaravone, over 60% of the treated mutant fish survived up to 21 days. Remember, the untreated ones were all gone by day 10 or 12.
Wow, okay, that's a huge difference in survival in the model.
It really underscores the potential impact.
Yep.
And it wasn't just survival. They also saw significant improvements in motor function. The drug treatment greatly reduced the proportion of fish showing those poor movement responses in the swimming tests.
So they were moving better too.
Yes. And importantly, to confirm the drugs were likely acting on the underlying problem, they measured those autophagy markers again, like LC3. They found that levels were reduced in the treated fish, suggesting the drugs were helping to sort of unblock or alleviate that disruption in the autophagy pathway.
That sounds incredibly promising. Finding compounds that improve survival, improve movement, and seem to be hitting the core cellular pathway. That's got to be encouraging for a disease with 0 treatments.
Absolutely. And one of the senior researchers, Dr. Alexander, was quoted emphasizing this point. He said something like, "The fact that multiple autophagy modulators ameliorated aspects of the phenotype supports an important role for autophagy in the disease process and lends confidence to the validity and potential translatability of the findings to patients." Basically, finding more than one drug that helps makes you more confident that targeting this pathway is the right strategy.
Okay, so really exciting leads discovered using this fish model. But, as you mentioned earlier, no model is perfect. They have limitations, right? The study pointed out some important caveats.
Yes, and it's so important to keep these in mind. Science is about understanding the nuances. While the zebrafish model was powerful, they were clear that, as we discussed, the disease severity in the fish is much more pronounced than in most human cases.
Okay.
Also, zebrafish muscle structure isn't a perfect match for human muscle. The way the fibers are organized is a bit different.
And those vacuoles, the hallmark structures in human muscle,
Right.
they didn't look exactly identical in the fish.
Yeah, that's right. The ultrafine details of the vacuoles observed in the fish muscle didn't perfectly replicate everything seen in human biopsies. The researchers suggested this could be a species difference, or maybe related to the fact they were looking at very young fish larvae. Perhaps older fish, if they survived, might develop features closer to the human ones.
And then there was that really interesting finding about the different organs, the liver versus the muscle.
Yes. This was a fascinating and potentially very significant nuance. Remember how the drugs Edaravone and LY29400002 improve survival and muscle function?
Yeah.
Well, they did not improve the liver problems, the impaired bile flow, the fatty liver signs. Those persisted even in the drug-treated fish.
Huh. So the same drugs helped the muscle issues but did nothing for the liver issues, even though both problems stem from the same original VMA21 gene defect.
Exactly. It's a bit of a puzzle. It strongly suggests that even within a single genetic disorder, the precise way the underlying defect causes trouble might differ between organ systems. Muscle and liver might be affected through slightly different downstream mechanisms.
What does that imply for treatment then?
Well, it could mean that a single drug might not be enough to treat all aspects of the disease. You might need different approaches, or maybe combination therapies, specifically targeting the problems in each affected organ. One strategy for muscle, perhaps another for the liver.
Wow, that adds a whole other layer of complexity.
Ah.
Okay, and thinking about the drugs themselves, Edaravone and LY294002, were there safety things to consider?
Yes, definitely. Edaravone has a bit of a head start there. It's actually already an approved drug used clinically for other conditions like ALS and certain types of stroke.
Oh, okay.
So it has a known safety profile in humans, which is generally considered favorable. That makes it a more straightforward candidate for further research aimed at potential human use for X-linked myopathy.
And the other one, LY294002.
That one's a bit different. LY294002 has known side effects that would likely make it unsuitable for long-term treatment in humans.
Ah.
But its value here is immense because it acts on a specific cellular signaling pathway known as the PI3 kinase AKT pathway. The fact that inhibiting this pathway helped the fish strongly suggests this pathway is involved in the disease process and could be a valid therapeutic target. So even if LY29402 itself isn't the answer, it points researchers towards developing or finding other safer drugs that target that same PI3 kinase AKT pathway.
I see. So LY294002 isn't really the drug candidate itself, but more like a crucial clue, highlighting a specific pathway that future drugs could aim for.
Exactly right. It opens up a whole new avenue for drug discovery specifically for this disease based on mechanism.
Okay, so taking all this together, the creation of this first ever functional animal model, the identification of promising drug leads like Edaravone, understanding the important limitations and complexities like the organ differences, where does this leave us? What's the big takeaway and what happens next?
I think the most critical achievement here is establishing the zebrafish model. It's the first functional animal model for X-linked myopathy with excessive autophagy, period.
That alone is huge.
It really is. It replicates many key disease features and, crucially, provides an essential tool for the whole research community. Now they have a platform to work with, especially for screening more potential therapies much more efficiently than before.
It basically unlocks a door that was previously closed.
Yes. And in terms of next steps, the researchers laid it out pretty clearly. They need to take these promising candidates, especially Edaravone, given its safety profile, and also look for other compounds targeting that PI3 kinase AKT pathway and test them in other systems. That means mammalian models, like mice specifically engineered with VMA21 mutations, and also, very importantly, in cell models derived directly from patients. See if the benefits hold up in human cells.
Right, moving closer to humans.
Exactly. And they'll likely keep refining the candidates they have and maybe even use the Zebrafish model to screen much larger libraries of compounds now that they've validated the system.
This really does feel like a significant step forward, maybe even a turning point for understanding and potentially, finally, treating a disease that affects such a small number of people across the entire globe.
I agree. It's a powerful example of how dedicated research, sometimes using creative or less conventional approaches like this Zebrafish model, can yield absolutely crucial breakthroughs. Even for these ultra-rare conditions where making progress is just incredibly difficult, it offers tangible hope.
It is pretty amazing to think that a potential breakthrough for a rare human genetic condition could start with watching tiny, transparent fish larvae swim under a microscope.
A potent reminder of where insights can come from.
Yeah. So this deep dive has really traced the journey, hasn't it? From identifying a single faulty gene in a handful of patients to engineering a fish model that shows many of the same problems, and now pointing towards actual compounds that might help.
And along the way, it's shown us something profound about biology, I think. How one genetic defect can ripple outwards, causing distinct problems in different parts of the body like muscle and liver.
Right, the drug helping one but not the other.
Exactly. And perhaps surprisingly, fixing the problem in one place might need a totally different strategy than fixing it somewhere else, even though it all stems from that same initial genetic typo.
That really does make you pause and think about the sheer complexity involved, doesn't it? What is that kind of variability, different organs needing potentially different solutions for the same genetic disease? Tell us about the intricate nature of these disorders and maybe about the future of treatment needing to be incredibly tailored and specific, something to definitely mull over.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.